So now we'll start our event this evening, and it gives me great pleasure to hand over to our Chief Executive, Dr. Robert Parker, who will introduce the event. Thank you.
Thank you, Pauline, and a very warm welcome, everybody, to our wonderful Chemistry Centre here in Burlington House. I'm delighted you could join us this evening for what's sure to be a fascinating discussion. This event, as Pauline said, is being broadcast live, so I'd also like to welcome our audience who are watching around the world. Today marks the first full day of our activities celebrating Chemistry Week, where this year's theme is health and how chemistry can have a positive impact on our lives. Earlier this afternoon, we had 100 16 to 18-year-olds in this library at a session of I'm a Scientist, Get Me Out of Here, with 5 early career scientists being quizzed by them at the front and them voting, all the young students voting on who was the best at the end of the day. That was a really, really good and interactive event. This year, Professor Dame Sally Davis, the Chief Medical Officer for England, has highlighted antimicrobial resistance as one of the major threats facing our society. It's a global problem, and fighting it will require global collaboration. New science is essential to this fight, and chemistry will have a very crucial role to play. Chemists will help to improve our understanding of resistance, help create the next generation of life-saving antibiotics and other drugs, and provide rapid ways to diagnose infections so that the right treatments can be delivered to patients at the right time. That's why we, the Royal Society of Chemistry, have organised this discussion this evening. As the world's leading chemistry community, we're working to shape the future of the chemical sciences for the benefit of science and humanity. We're committed to supporting research and driving innovation that will limit the impact of antibiotic resistance. But of course, chemists can't achieve this alone. If we're to meet the global healthcare challenges posed by antimicrobial resistance, it's vital that chemical scientists work in partnership with other scientists in academia and industry and engage with policymakers, clinicians and the wider public with new ideas and scientific advances. Our panel of experts will cover many of these topics and more this evening, with experience ranging from clinical medicine to policy making and antibiotics research and development. We've no doubt that they'll provide fascinating insights and unique perspectives into the problems we're facing and the solutions we have to find. We're extremely fortunate this evening to welcome Dr. Michael Mosley as our chair for this discussion. Many of you will recognise Michael from his extensive work on television. Less well known are other aspects of his exciting and varied career. Michael was born in Kolkata, India, before moving to the UK. He studied philosophy, politics and economics at Oxford and went on to join the exciting world of investment banking before deciding that he really wanted to study medicine. Having qualified as a doctor, he realised that actually a career in medicine wasn't for him either, and so began what is has now a 25-year-long career as a science journalist, TV producer and presenter. He's made many scientific documentaries and writes for The Times, The Independent and The Mail on Sunday. His work has won numerous awards, including a BAFTA and 2 Emmy Awards. He's also been recognised by the British Medical Association as their Medical Journalist of the Year. Michael, we're very grateful you could join us tonight. Now, I'm sure we're all as keen to hear what the panellists have to say, so without further ado, I'll leave you in charge of Michael. Thank you very much.
Thank you very much. Good evening. Hi, all. Thank you. In a moment, we're going to be talking to the panel. I thought I'd just give you a little view from the less professional end of the circuit. I've recently done a series called Pain, Pus and Poison, which is a celebration, if you like, of the extraordinary voyage we have been on for the last 150 years to first identify the role that microbes play in infectious disease and then try and do something about it. I mean, it is amazing that it's 70 years since penicillin was first used, and it's only really been 150 years since we recognized that microbes, their potential to cause disease. And chemists have played a huge role in the understanding of microscopic, the relationship between microscopic life and infection, and indeed in the treatment of it. I like the fact that Louis Pasteur was a chemist, and I particularly like the fact that it all kicks off with the English complaining about French wine. Did you know that? That's where it all begins. Basically, the English complain about the quality of the claret which is coming over, and there is such horror amongst the French that they employ chemist Louis Pasteur to find out why so much of this wine is going off, and that's what leads him to investigate the role of microbes, and indeed leads to pretty well our earliest understanding of the role of microbes. And then that goes on, and you have Ernst Chain in Oxford, another great chemist, who helps to isolate the active ingredient in penicillin. So hurrah for chemists, I would say. I personally have a huge debt of gratitude to the people who worked on those things, because I was born in India. My mother said I would have died at the age of 2 if it hadn't been for antibiotics. So I would not be sitting here. But the challenge obviously is the future. What do we do next? Because the title is Beating the Superbugs, which sounds optimistic. And I'm hoping that our panel is going to be a little bit optimistic. I suspect we're going to hear some horror stories, but as I said, I'm hoping for a little bit of a ray of sunshine from them as well. We have Dr. Nick Brown, who is a consultant medical microbiologist at Addenbrooke's Hospital in Cambridge, and he's been closely involved with the Antibiotic Action Campaign. Next to him, Professor Barry Cookson, who is medically and dentally qualified. And he is also a medical microbiologist and was for many years director of the Laboratory of Healthcare-Associated Infection. And he's currently a professor at University College. And Professor Chris Schofield, who is head of organic chemistry at the University of Oxford, where his research group works at the interface of chemistry, biology and medicine. And his work has opened up new fields in antibiotic research, oxygen sensing and gene regulation. So they're all going to be talking in turn. Then we would love to hear your questions. When you have a burning question, please stick up your hand and the microphone will come to you so everyone can hear. Before we go to the panel, though, we would like to hear from Professor Dame Sally Davis, the Chief Medical Officer, who is coming via something electronic, I think.
Good evening, Michael. Hello, everyone. I'm delighted that you're having this discussion about antimicrobial resistance. Of course, microbes take in everything that's infective. It takes in bacteria, and that's antibacterial resistance we're worrying about, but fungi, viruses, everything. And the most important antibiotic, or best known, was penicillin. And you know that Alexander Fleming, when he received his Nobel Prize, predicted antimicrobial resistance and that deaths would follow. It's a growing problem. Now, 25,000 people die in Europe every year from antibiotic resistance, exactly the same as road accidents, and the human cost and societal cost is immense. Indeed, one baby or young child dies every 5 minutes in Southeast Asia, and the Americans have a conservative calculation of 23,000 deaths a year. So why am I worrying about it at this time? I'm worrying because we haven't had a new class of antibiotics since 1987. And until now, when a bacteria or a bug developed resistance, there was always another drug in the cupboard, so we could save lives, and it wasn't that serious. But with this empty pipeline, we don't have new drugs. We're going to have to discover new drugs. We're going to have to work out how to live with antibiotic resistance, or we could go back to a pre-antibiotic era where just under half of us would die of infection. We don't know the size of the problem because we don't have good laboratory testing and surveillance across all the world, and we do know that this is one health issue, and by that I mean antibiotics are used in animals, in some countries just tipped into the animal or fish feed in order to promote growth cheaply. A third of countries have no regulations about that. And also people pick up antibiotic resistance in one country and take it to another. Meanwhile, we aren't as good as we were in Victorian times at hygiene. Florence Nightingale taught us a lot, and we've lost some of that. We need to rediscover it so we don't move bugs around. But this is a growing problem and the drugs don't work anymore. And we need everyone to work together to get a solution to this so that we can go on with modern medicine. Without antibiotics, people won't be able to have cancer treatments, transplants, even routine hip surgery. A replacement hip needs antibiotics. So this is a grave threat to civilization as we know it. And actually our government's going to put it on the risk register next door to terrorism, pandemic flu and climate change. It is that important to all of us.
Right. Dr. Nick Brown.
Good evening. I've been asked just to give a couple of minutes to introduce antibiotic resistance from the perspective of a practicing clinician and a hospital microbiologist. As we've heard, antibiotic resistance shouldn't surprise us. The bacteria have been around for hundreds of thousands of years, millions of years, and have been exposed to naturally occurring antibacterial agents and have developed mechanisms to get around that, to survive. So resistance will occur, as Fleming predicted. But the issue, as Dame Sally has just emphasised, is that although there were 14 classes of antibiotics developed up to 1968, we've only had 5 classes of antibiotic since then, and arguably only 2 that have been therapeutically useful. And so the antibiotic pipeline for development of new agents has really dried up. So this isn't an acute issue just now. It's not just that we need new antibiotics, but we need to create a way of developing antibiotics into the future to sustain our antibacterial therapy for our grandchildren, their grandchildren, and so on. So what does antibiotic resistance mean to me? Well, for a clinician, most of the antibiotics that I prescribe are prescribed without knowing what the infective organism is. At the point that I sign a prescription or a colleague signs a prescription, we know that the patient has an infection, but we can guess, but we don't know exactly what the organism is, and we certainly don't know whether it's due to an antibiotic resistant organism or not. And therefore it's empiric treatment, and increasingly we're finding that our initial choice of antibiotic is wrong, that the organism is resistant and therefore antibiotic treatment fails. And there are lots of data to show that if you get that initial treatment wrong, then people do badly. Either ultimately, the worst case, obviously, they might die, but they might stay in hospital for longer. The costs of treatment are by far escalated, and their outcome generally is much poorer. So what, from my perspective, do we need to do about this? Well, firstly, others are going to talk about stimulating the pipeline for drug discovery, but also a big part, I think, is also protecting what we've got. We know that resistance will occur, we know resistance can spread, but if we use antibiotics better, then perhaps we can slow the rapid spread of resistance that we've seen over the last couple of decades. Thank you.
Professor Coxon.
Good evening, everyone. I'm assuming that everyone's read the UK strategy that Dame Sally has published recently. I've been asked to say just a few things about policy. Now, this is a multifaceted, very, very difficult issue, and there's no quick fix here. But I just wanted to cover 3 particular areas. The first one, in terms of priority, is strengthening international collaborative working. Now, you'll have seen that Dame Sally has aligned the UK strategy with the ECDC strategy exactly. Perfect fit. And that's totally appropriate. If you go way back to 1999 and look at the WHO, the World Health Organization strategy, it's very similar. There's one area where we have completely failed, and that's advocacy. And I think we have a wonderful opportunity, because Dame Sally is going to be the WHO UK representative next year. And I'm hoping that, rather like Liam Donaldson, her predecessor, who was a champion for patient safety, and his predecessor, Kenneth Kalman, who was really one of the major people who said clinical governance has got to be progressed in this country after the Bristol cardiothoracic deaths. And he really drove total quality management in this country. So we have a wonderful opportunity of Dame Sally being the champion for this cause. I'm a bit of a half-full cup guy, but I really am positive about that. Now, the two major weapons that we have in this fight are antimicrobial stewardship, but also infection prevention and control. And it's just as important, if not more, arguably. And it's been very much the Cinderella in this particular fight. And I was pleased to see that ECDC and Dame Sally have both got it as #1, actually, in the strategies. Now, we're not just talking about healthcare infection control, washing your hands. We're talking about better waste management, appropriate cleaning of toilets, clean water, which much of the world has not got, and kitchen cleanliness. How many of you have seen in a cookery program on television them actually washing their hands? How many of you are aware of how to keep raw food separate from cooked food in the kitchen? I won't ask you to put up your hands, but this is really important. And the other area is animal husbandry, and I think I'll leave that because there may very well be experts in the audience on animal husbandry, and we can cover that in the questions. I'll also leave policy aspects of encouraging new antibiotic and new treatment developments to after the next speaker, and perhaps some questions from the floor. Thank you.
So it's great to see so many people here tonight. I hope there's a few non-experts as well. I guess when you get to a certain age, almost everyone knows somebody who's actually died of an antibiotic infection. So this really matters to us. The first thing I'd like to say is, I think in terms of addressing new antibiotics, new pharmaceuticals, this is a solvable problem. It's a problem that will never be fixed forever. We'll have to continue to do it. But it has been solved once. So, for example, the mechanism of resistance that Fleming predicted was addressed in the pharmaceutical companies, and I should emphasize, primarily in the pharmaceutical sector in the 1970s and 1980s, in fantastic science. They developed a combination of therapies where you had a penicillin and a compound that neutralized the resistance agent. And these, one such formulation is Augmentin, that many of you will have had. These were tremendously successful medicines and are still used to this day. But we need to get back to that spirit of innovation. And the truth is, as a community, we failed to develop new antibiotics and antibiotic anti-resistance agents. And this is absolutely not the fault of the pharmaceutical industry, I should say. They haven't been incentivized to do that. It's a fault of the whole community, and we need to address that problem collectively. It's a political problem now, how to solve this problem. So just say a few words about the role of chemistry in antibiotic development. Chemistry is crucial in terms of making new compounds to test as antibiotics, validation of new targets, and there's a question as to whether or not we really need new targets. I'll come back to that. And in developing new diagnostic methods. And it isn't just chemists, it's chemists in collaboration with biochemists, biologists and clinicians as well. It's very much a team effort, and that's exactly what the pharmaceutical industry did in the 1970s and 80s. So there is a problem here. I don't know how to solve it, but it needs to be addressed. It can be addressed. At the moment, the commercial sector isn't solving the problem, so something has to change. It won't be changed by funding a few research projects in universities as well. This in my opinion, requires a large-scale effort. So what might we do? Well, actually, in the early days of the development of the penicillin story, it wasn't just Fleming, it wasn't just Oxford, it was Fleming, Oxford, the United States, and it was a genuine public-private partnership that occurred during the Second World War, where companies and academics worked together, and it was a huge effort to make penicillin clinically viable. And that's the kind of thing we could do again. It's either doing that, in my opinion, or incentivizing the private sector. And to incentivize the private sector will probably involve legislation, and not just in this country. And that might take too long. So I'll finish there and
thank you. Right, so lots of provocative stuff. There's a big problem and possibly a big solution. I'm going to go to the floor in a moment and ask questions, but we have two I'd like to take. One is from Professor John Watson, who is the Deputy Chief Medical Officer.
Thank you very much for this opportunity. Firstly, I'd like just to pick up on a comment that Barry Cookson made about the international work and the importance of international collaboration, and I certainly couldn't agree more. We've had strategies, quite a number of strategies over the years. What's different about this? And I think one of the big things that's different about this strategy is both the recognition of the importance of getting that genuine international collaboration and the energy that people like Sally Davis, the Chief Medical Officer, is putting into that personally to really push and push and push again in various international fora, WHO, European Commission, in various discussions with other countries, to push them to also work on this and make things different. That's the comment. The question I'd like to put to the panel, if I may, really is to begin with by agreeing with your emphasising the importance of developing new antibiotics, but suggesting that in doing that you've neglected other bits of what the solution to this may be, which relate to aspects of the prevention and other approaches to the treatment and management of this. In relation to prevention, infection control, Barry, you mentioned, I think is so very important, but there are other ways that we could manage without antibiotics. Further use of vaccines so that people don't get those infections in the first place. And I wonder, Barry, would you like to comment on that as part of the strategy? But secondly, when it comes to the management of infections, we've heard in recent weeks about some fantastic novel approaches which really need some additional oomph behind them. The use of viruses, bacteriophages, targeting specifically some of these bacteria, and I wonder whether panel members would like to comment on what they think is the potential for those going forward. And there are other novel approaches of that kind that don't necessarily involve the use of antibiotics. For example, work that's been done on better understanding and utilising the microbiome, those bugs we already have within us and are, for the most part, our friends rather than the enemies to be fought in a war. And I wonder whether you'd like to comment on that too. Thank you.
Thank you. So, Professor Cookson.
Well, shall I take phage and vaccines and you take, yeah? Okay,
Carve it out between
right.
them.
So I'll start with the easier one, that's phage. When I was directing the Laboratory of Healthcare-Associated Infection in the Health Protection Agency, as was, I was specifically asked to explore using phages. Now, there are two countries in the world, Georgia and Russia, both of whom use bacteriophage.
Excuse me, sorry, does everyone know what phages are?
Phage means eat, and these viruses actually destroy bacteria, but they're specific for the particular bacterium, so Staph Aureus, for example. The bacteriophage that sticks on Staph Aureus does also stick on your skin staphylococci that you have on, everyone in this room has, but it won't kill them, but it will kill the Staph Aureus. So it's called bacteriophage, bacteria eating. Now the problem is that you have to have a cocktail of bacteriophages. You have to, so if you have a Staph Aureus infection, you have to have a cocktail of bacteriophages so that it can kill many different types of Staph Aureus. That's the first problem, because the USA in particular just completely refuses to clear any sort of multiple preparation like that, and I think I'll leave that to the regulatory people in the audience. The huge problem I had when I was asked to look at this was when we spoke to the people using it, we worked out that for the UK we would need the whole of our institute, which had about 350, 400 people, just to cover the bacteriophage therapy in this country. They use it for infections which are failing on antibiotic treatment, and once they've started treating, you have to monitor literally every week, and when resistance occurs, you have to start developing a new bacteriophage. So it is not an easy area at all. There are some interesting new chemical developments. People are even thinking about injecting bacteriophages for systemic treatments. And there are ways in which they've chemically altered the bacteriophage so that it's not gobbled up by your liver and your reticular endothelial system, because it rapidly disappears if it's injected into you. So can I now move on to, the other thing that we've done, in fact we've got a patent on this. I mentioned that bacteriophages non-specifically stick to all staphylococci. That was something we thought was correct, but we actually proved it by sticking photochemical agents onto the bacteriophage and then using them to target the bacteria and then lighting them up with a UV so they destroy the bacterium. So we've actually used bacteriophage to carry the chemicals onto the organisms. And that's a little bit like the nanotechnology approaches that we're hearing about now, where we're developing molecules which will carry drugs into specific sites.
So the hope was that viruses would be used to kill bacteria, but that hasn't worked out terribly well.
It can be done. It's particularly useful when you have got biofilms, particularly polysaccharide biofilms, which a lot of hospital infections are, on veins and bits of plastic. But resistance occurs too quickly, and it's far too expensive, and really it hasn't got much of a future.
Oh dear. One down.
Yeah. So
moving on.
Shall I move on to vaccines? Because that's your area of expertise, John. You're being very modest.
Vaccines, yeah.
But okay, vaccines. Now, because there's not been too much of a market until recent years in hospital infection, it really was a Cinderella subject, actually, rather like infection prevention control. It wasn't even on the business plan, the first business plan of the Public Health Laboratory Service in 1993. So only in recent years has it become a sort of a sexy area. The only country that really was doing interesting work in this area was Israel, where they had large numbers of burns patients with Pseudomonas aeruginosa. They did use phage therapy, and it was quite successful actually, but better than that was vaccines. So they developed a Pseudomonas aeruginosa vaccine. There is another area, Acambis have been bought by Sanofi Pasteur, and they have got a Clostridium difficile toxin, which is in its phase 3 at the moment. And the other area is Staph Aureus, where there are 4 companies that have developed vaccines, but unfortunately 2 of them have failed, and there's a huge debate going on in the literature. Staph Aureus pathogenesis is very complicated, and there's a huge host component, a cell-mediated immunity component, which people don't realise, and also they're intracellular, they get inside cells as well, rather like salmonella and TB. So there's a huge challenge. It's likely that a successful vaccine will have several targets. And I've noticed that another approach is being funded in Europe in the IMI call, and that is a passive immunoglobulin approach to the alpha hemolysin of Staph Aureus. So it's beginning to get better than it was, and you're absolutely right, it's on my list of very, very high priority that we develop these vaccines, because we're gradually running out of antimicrobial options.
So you're quite optimistic about vaccines at the moment?
I'm more than optimistic. I really think that, that's where we've got.
Because I was very surprised when we were talking earlier, and previously they used to dump a lot of antibiotics into fish. Fish used to consume huge amounts of antibiotics, and now apparently in the UK they're almost all vaccinated. So if you buy fish here, you're not going to be dosed up with antibiotics. Is that right?
Yes.
Is that true of cattle as well?
Is there an expert in the audience on aquaculture? I mean, I don't want to start. There's someone in the audience who knows more than I do, which is not difficult.
I didn't even know they vaccinated fish.
So there are about 6 fish pathogens, all of which have got vaccines. It is illegal in this country to use an antibiotic in the fish industry. They all use vaccines. China has got some vaccines, but they're largely using antibiotics. Chile use 7 times more quinolones for fish than they do for humans. There are huge amounts of antibiotics being used in the fish industry, and there was a survey done just a year or 2 ago of 760 people involved in the industry all around the world, and the astonishing finding was that in the USA, where it's banned, and Canada, where it's highly recommended you do not do it, between 60 and 80% of the people in those countries were actually using antibiotics in the fish industry. I think largely through laziness, because it isn't the easiest thing to do that, to vaccinate fish.
So the good news is that the vaccines are kind of there, they're spreading, they're effective. The bad news is some people prefer just to chuck a handful of antibiotics in to treat it.
And huge amounts. They reckon 70% of the prescribing in America is in the non-human area, and in China it is 50%. And they reckon at least half of that is in the fish industry. And the other point is that...
Chinese fish. Oof.
Yeah. The other thing is that when you compare the spread of antimicrobial resistance in fish to either humans or animals, it's much, much faster. It spreads obviously through the water, but also it's been shown many, many different species, the same plasmid can go from one to the other.
I'm going to move on. Professor Schofield. You stand accused of ignoring a huge range of other particular ways of treating.
So I think the potential, particularly of vaccines, is enormous, but you've got to give us some kind of timescale on which they're going to be replacing small molecule therapy. I think in my opinion, and that of Sally Davis as well, they're unlikely to replace penicillins and related antibiotics on a timescale of 5 to 10 years. And we notice that resistance is on the risk register now, so we need to consider what would happen if there was a major outbreak of resistance. Say all the penicillins and related compounds stopped working in a 2-year time frame. What would we do? In my opinion, that's unlikely to be vaccines. It's going to be develop new small molecules via the tried and trusted procedures that have worked previously. And right now, the concern is that in this country and large parts of the world, there isn't the capacity in terms of pharmaceutical science and medicinal chemistry to address that problem. And for me, that is a serious concern for the country.
Nick?
I agree with that entirely. I mean, I think the issue with regard to the drug development is that in a way, we've picked off the low-hanging fruit already. The penicillins, some of the related compounds that have been developed in the early days of antibiotic therapy were by and large natural products that could be picked from the environment and then developed, and then subsequently developed to expand the armory of antibacterial therapy. What we're finding now is that it's actually much more difficult to develop these compounds, identify them in the first place, and then find those that have got clinical utility. And I think that will be the challenge for the immediate future, as has been said, rather than changing tack entirely to replace these compounds.
Okay, we will come back to these things, I'm sure. Animal resistance, the development of new drugs. One final question from Professor Sally Davis before we start.
If the cupboard's bare with antibiotics, and there are very few in the pipeline, that means we haven't incentivized research, and also that it's quite difficult to do that research. What do we need to do to get good research translated into drugs for patients?
Okay, shall we start with Chris?
Well, to repeat what I said before, what we have been doing hasn't been working. And in my opinion, there is perhaps not as much research and development in this field as you might think. If you compare the amount of global pharmaceutical activity, say, on anti-cancer compounds, where there is an incentive to develop drugs or respiratory agents, compared to that on antibiotics, the amount of research on antibiotics is very small. And to repeat, that isn't the fault of the pharmaceutical companies. It's something that society has to fix. Now, one possibility would be people work together better than they have been. And we need to think how we fund this. This is expensive to develop antibiotics. We're talking, 50 to 100 million pounds per new antibiotic, or maybe much larger sums of that if you take into account clinical trials. However, there's one important thing about antibiotics you need to know. The risk in clinical trials, providing you choose a validated target, is relatively small compared to most pharmaceutical enterprises. So the risk in clinical trials for, say, something like pain or depression is enormously high. And I don't think it's right that the public sector can take that risk. But in the case of antibiotics, it's still high, but relatively low. So I do believe that antibiotics are one class of pharmaceuticals that could be developed in the public sector. I'm not saying they should be, but I believe they could be. There is the funding capacity within, for example, the NHS budget to do that, if we wanted to do it.
Barry.
Yeah, I've got some figures here. I'll just introduce it by saying that a friend of mine working in the pharmaceutical industry said to me that investing in a new antibiotic is a form of madness. And the reason he said that was that they are incredibly expensive to produce. And you're fighting trillions of microbes. They've got all sorts of mechanisms that they can develop to produce resistance. And they're incredibly clever. Vancomycin resistance, we said, was going to be virtually impossible, didn't we? They assembled 7 different genes from 4 different species on a particular genetic element, which altered their cell wall to make them resistant. I mean, it's unbelievable. And the other point, which we're actually now, I think, going to start using against them, if I took a swab, I'm a dentist, if I took a swab of your gingival crevice in this room and I did a universal PCR against the ribosomal element in there, I would only be able to identify about 5% of the organisms in your mouth in that crevice. Now, what they're beginning to do is they're now, if you extrapolate, perhaps 90% of the bacteria we cannot grow. So what they're doing is they're taking the DNA in the environment, cloning it in E. coli, and expressing it in another organism to look at novel therapeutic agents. I think this is a really fertile area. But coming back to why we've got this problem. I went on the web and I found a couple of sources actually. Antibiotic revenue, 19.6 billion in 2009, is gone down to 16.4 billion in 2019, they reckon. Total sales have fallen about 1.6% per year since 2005, and that will increase to 1.8% to 2019. Between 1997 and 2006, reasons not given, antimicrobial prescriptions have dropped 13%. Now,
isn't that a good thing?
Well, that could be a good thing. That could be a good thing, but not if you're actually trying to sell them.
Not if you're a pharmaceutical company.
I tell my students, if I won the lottery and I had £1 million, the last thing I would do, and I was told I had to invest it in one drug. Yeah. I don't know how many people in this room would have put it into an antibiotic. I certainly wouldn't. Now, the big problem is that the patents only last 20 years after you file. So industry is wanting to extend the patents. Now, you can do that under an orphan drug approach, and that has been used in cystic fibrosis with tobramycin. So coming back to what you're saying, we could discuss changing that policy. You can, in the orphan approach, have tax incentives. You can subsidize financially their research. But the really important area that we discussed way back in 1999 and has now come to reality is have private-public partnerships. And there's a huge one started in Europe, the IMI. It floundered in the first 2 years because they only gave 20% overheads to the universities and increased it. But if you go on the web, there are several new projects underway, and I've just been asked to help sort of referee one or two of them. And so that is yet another example of how we can incentivize industry.
Nick, are you feeling optimistic?
Not as optimistic, I don't think. I mean, I agree entirely what Chris and Barry have been saying, but I think we also have to think about why drug companies aren't taking up this challenge. And many of the big drug companies are actually leaving this field altogether. Some of them that used to be very big players in this country and have huge research establishments have actually left the country entirely. And Britain, that used to be a leader in antibacterial drug development, really has very few scientists who've experience now of having taken a drug to the market. We have lots of people working on it, but in terms of that experience. And I think we ought to think why that is, and some of the points have been touched on already. But if we compare, say, an antibacterial drug to an antidepressant or a statin for cardiovascular disease or an antihypertensive, these are drugs that, although they might be quite cheap individually, they're drugs that, by and large, members of the population are going to take for many, many years, whereas we're saying to people with infections, well, actually lots of infections are viral, you don't need an antibiotic at all, or if you do need it, you're going to have it for 5 to 7 days. It's not the same market as we've got for many other drugs. Plus, of course, we're not actually treating the individual. We're treating a third party, the bug. And drugs that are designed to kill bugs often aren't as good for humans as some other drugs that are designed specifically for that purpose. So actually, despite what Chris was saying, that the attrition rate in drug development for an antibacterial agent is far higher than it is for some other drugs. And a figure that was quoted to me last week was that actually of 27 prospective agents, only one will make it anywhere near a phase 3 clinical trial because of issues such as either rapid emergence of resistance or toxicities, which will mean that actually clinically it's not terribly useful. And then a final point is that we do have a candidate drug. It's gone through early trials, it's promising. Then to get it through the regulatory framework to actually get it to market is now an area that is fraught with difficulty for companies, and the regulatory framework is different depending which side of the Atlantic you're on. One of the areas that is causing some consternation at the moment is that the basic requirement to get a drug marketed is that you have to show that it is not inferior to the agents that you're using at the moment, which sounds fine, but clearly, ideally, we'd want to show that a drug was better than the agents that we're using at the moment. But the non-inferiority studies are much easier to do, they're much cheaper, you need fewer people to do them in. But then, of course, at the end of the day, when the drug does get marketed, all the company can say to people who are prepared to prescribe is that I have a new agent here, it's a lot more expensive than the one you're using at the moment, and all I can tell you is it's not worse than what you've got now. So
it's not a good selling point,
so
is
it's
it?
not a terribly good selling point.
No.
So immediately it will be relegated to third or fourth choice. So I think there are a number of issues to do with the actual development process, the regulatory framework, etc., that need to be considered as well.
Right.
So Nate, just to come back on that, I totally agree with your points about regulation. That's hindering the antibiotic drug development. But my comment about the clinical trials was referring, in my case, to validated targets. And maybe one of the issues have been people have been pursuing novelty, both in academia and the pharmaceutical industry, rather than utility. I think you absolutely have to start from a clinical need. And if you're meeting an unmet clinical need, by definition, it's going to be better than what's out there. And then work back to your molecules and your targets from that unmet clinical need. I think of those 27 you mentioned, I bet most of them were new targets. And I just want to mention one other issue where chemistry comes into play, and that's cost of goods as well. So antibiotics, you can't charge very much for them as well. And some of them are quite complicated molecules, things like penicillin and erythromycin. It's really difficult chemistry to make these structures. And that's where chemical synthesis comes in. You have to make them for a few tens of pounds per kilogram. And this is arguably the most difficult type of chemistry to do. And some of the triumphs of 20th century chemistry have been to make some of the antibiotics we all use on a large scale by so-called total chemical synthesis. But a number of antibiotics have failed on that issue as well, cost of goods.
Just too difficult to make.
Well, no, but chemistry's, we're getting better at chemistry and synthesis.
Hurrah for chemistry.
I think if penicillins were discovered today, we probably wouldn't ferment them. We'd probably make them by total synthesis.
Right. They'd never get through. They kill guinea pigs.
Well, that's a
anyway, time to open it up to the floor. Questions, please, on any aspect of whatever you've heard. Gentleman over there, wait for the microphone. If you could introduce yourself and either address the panel or a particular person you wish to ask.
My name's Peter Freeman. On a note of cynicism, the lady who came on mentioned the priorities of the government, and they aligned antibiotic resistance with terrorism and climate change. I sincerely hope we're getting better at dealing with antibiotics than we are with dealing with climate change.
Who would like to take that one?
I think antibiotics can be done. I'm not sure about climate change, though. That's the difference.
I mean, the point is well made. You know, the regulation is going to be a massive challenge here. If you go on the ECDC website, there is a roadmap for the next 5 years, and a lot of it is addressing regulatory issues. But that's just Europe.
I think, likewise, the point also rings true in that this has been discussed for many, many years. In the UK, the House of Lords Science and Technology Committee undertook a review that was published in 1998. Sorry. Yes, but so it's about 15 years ago. Anyway. And so even after that, there was an action plan and a strategy to combat antibiotic resistance. But many of the things that we're talking about now are exactly the same as they were then. I think things, on the positive side, I think things have moved on quite significantly, particularly on the international aspect that the WHO has been mentioned already. But there are a number of global alliances that have discussed this at significant length. And I think, I just get the impression that there is a renewed impetus to try to get something started, at least now, that perhaps wasn't there before.
I was going to say, because one of the things that the question of global warming brings up is the question of, is there likely to be a technological solution? I mean, do you think it is possible to conquer the superbugs, as the title suggests?
No, absolutely not. I mean, this is a continuing battle. But the problem at the moment is that the armoury is rather empty. I just want to say a couple of positive things as well. I mean, there are some rays of hope here. For example, AstraZeneca are hoping to introduce a new combination therapy to the clinic soon, which will be an antibiotic plus an agent that protects the antibiotic as well. That will be coming through soon. And if that makes it through to the clinic, that will be fantastic. For me, that will be one of the most important things in antibiotic therapy for several decades. But the question is, will they be able to commercialise this? We want to do that again and again and again.
Can I just get back to... Sorry, can I just,
yes, please.
Going back to the economics and trying to think outside the box, someone wrote a letter in response to the recent lead in the Lancet, pointing out, I think it was Costerton, basically was saying we are undervaluing antibiotics. Antibiotics are incredibly valuable as a drug, and so they should be more expensive than they are, which is an interesting approach. There are two areas where, one of which we're doing better.
So you're suggesting some form of economic rationing, by making them more expensive, therefore less...
And they're saying it's easier to do in the UK, where the patient isn't paying for them, or not directly.
Yes.
But in some societies they would be. But his point is that if you look at the value, they are incredibly
a straw poll, would you be prepared to pay more for antibiotics? You think that would be a rational approach? How much would you like to see them charge? Double? Triple?
I think at least triple, but you'd have to look at the particular condition that's being treated and for how long, with what antibiotic. You'd have to tailor the costs and society costs and so on. The econometrics are very, very complicated. The impact of a hip infection can go on for 20, 30 years. So you have to factor all these things in. There's one area where we're doing better in terms of chemistry. I was at a meeting, it was a closed meeting in America last year, and it was unbelievable. They were using crystallography and DNA methodologies, and it was that they were changing, that they knew exactly which part of the molecule was causing toxicity, and they knew which part of the molecule was causing the antibiotic effect, and they could alter one without altering the other, and then remanufacture the product. We haven't got into an animal yet, but that's the sort of thing I was impressed with. But there's another area which we are not good at. I'll just, and that comes back to that joke about a form of madness is investing in antibiotics. Predicting when and where and how important resistance is going to occur is a nightmare. One does all sorts of things in the laboratory to try and predict it, but time and time again, the bacteria prove cleverer than you thought.
Nick?
I don't have much to add on that subject, actually. Yeah.
Okay,
Yep.
fair enough, very honest. Question?
Oh,
Yeah.
sorry.
So yes, we'll come back to your question from the gentleman over there with the microphone, and then the lady over here,
Yes.
and then the gentleman over there. Hello.
My name's Anthony Coates from St George's, and I have a question for Barry and a question for Chris. My question for Barry is incentivisation of the pharmaceutical industry. Unbelievably important, and you've mentioned how low the unit price is. I mean, for example, antibiotics, $10. Some anti-cancer drugs, a few of them, $10,000, $100,000 even. So there is a huge gap, and that's why industry is not investing properly in antibiotics. The Infectious Disease Society of America has suggested something called limited population antimicrobial drug pathway, which basically means you do your trials, smallish trials, with drug-resistant organisms, and then you are allowed, as long as you just keep your treatment to drug-resistant organisms, to charge a high unit price. So I wanted to know what Barry thought about that, and for Chris, I wanted to know what he felt about stimulating universities to undertake research, and perhaps the research councils should get together in some sort of cross-council program. Perhaps there should be chemistry hubs. Here we are. Stimulation to chemistry by having chemistry at the centre of basic research into antibiotics, and the rest of us, microbiologists and so on, would gather around these hubs throughout the UK. I wondered what Chris might think about that.
Okay, so the first question is really, we're going back to econometrics and widening the topic. I mean, you'd be talking about tailoring your economics to the particular clinical situation, and now you're talking about drug-bug combinations. So it really is quite an exquisitely, it's an enormous area that you'd have to develop this. And I keep on thinking, what we said back in '99, perhaps the industry shouldn't be making antibiotics at all. Perhaps it should be a public, government-sponsored area.
Well, presumably the government could put up some money and say, okay, you make us the antibiotics.
They're already partnering industry in the IMI initiative, and America's got another huge partnership going on. And I can see kind of the way it's drifting is perhaps it should become purely public funded. But we will have to think about things you've mentioned, the drug bug indications, likelihood of resistance. Is it going to be reserved for resistant pathogens, that particular antibiotic, but also the clinical conditions, and looking at the society costs and the long-term costs. I mean, Dame Sally quoted figures in her strategy, but they are a vast underestimate, as several people have said. I mean, even the 1.1 billion, that doesn't include intensive care costs, it doesn't include expensive antibiotic costs, it doesn't include the cost of infection prevention and control. It's a vast underestimate.
Chris, are you going to be busy developing antibiotics sometime soon?
I think we have a representative of the MRC here tonight, actually, who may care to comment before I speak.
Yes, hi, I'm Des Orsh. I'm from the Medical Research Council. I'm the head of infectious immunity and the lead for stratified medicine there. Just to echo what Andy was saying, the research councils are taking this very seriously, and we are getting together at the beginning of December, informed by a number of workshops that each of the individual councils have had, which Chris was president at our one. And actually, we recognise the need that we have to fund across our own boundaries and across our own borders, and we need to bring chemistry into close partnership with bacteriology, also with the vaccine community, looking at immunologists, some of the, there are some really fundamental issues that I think chemists can address, and that really came out very strongly in a workshop we had back in the summer, and is in our report. But we are working as council, but we're extending beyond that. So it needs to involve the Technology Strategy Board, which in the UK is able to fund companies, particularly small companies. We will link up with the Department of Health. Clearly, they are very, very interested in what's happening in AMR. But also beyond that into Europe, there's a joint program initiative in antimicrobial resistance, which MRC are representing the UK on, and that will produce its strategic research agenda early next year. And that intimately is associated with the IMI. But we want to go beyond. We work with Canada, Israel, India, and China. So there's a lot of work ongoing at the minute. It does take money, and we do need to get that money together, and we need to get it into the right shape and format, so then we can start investing in the areas that experts such as Chris and Anthony tell us where we need to invest.
Are you including organisational behavioural change in that?
There is, yes. ESRC are looking at that, and also education of general public and medical practitioners.
So Chris, you're going to be putting in your grant?
So I think we need to be honest here. The track record of academics and making medicines is not terribly good. So in this country, as far as I know, there's one who's done it in recent years. That's Malcolm Stevens at Nottingham, who was the inventor of temozolomide, which is a really important cancer therapy. So what has been successful in this field is the commercial sector, but they haven't been recently. So we need to look at what they did. It is expensive to make medicines. You need a team of people, maybe 10 to 20 people per medicine, working for a period of up to 10 years per medicine in a focused way. Taking a relatively small amount of money and spreading it out over, this is the European way, over 10 to 20 groups, has not proven successful to date. Right. So we need to do something different. I think the MRC and the research councils are brilliant at funding basic research. So if you want to look into the fundamental science of resistance mechanisms, MRC, BBSRC are fantastic at that. But whether they're the right vehicles for making new medicines, new pills, if you like, I need to be convinced. I think it will need a change in ethos from those agencies. I think in the UK, from my perspective, a somewhat controversial thing, the agency that could do it, has the budget to do it, is the NHS. It could really do it. Now, whether it should just be the UK or an international thing, this is an open question. But for the politicians, the question is, if the penicillins and the erythromycins stopped working 2 to 3 years from now, what would you do?
So is this over to Professor Watson then? Are you going to open your pockets? Do you have access to money?
Money bags.
Thank you for that.
Sorry, I put you on that.
Personally, I have any access at all, but what I would say is that, in fact, the UK government, and further spurred on by Sally Davis, is indeed putting its money in its pocket. Of course, the MRC is part of the UK taxpayers' contribution, as is the NIHR, the National Institute for Health Research, which takes some of that work, but from the basic science, and then works on the translational work that moves it closer to the patient. So there's already quite a lot of work in research funded directly in this country, and in collaborative work through Europe, through the Innovative Medicines Initiative in Europe, through arrangements with the United States, through, I think it's called TATFAR, and I can't remember what the acronym stands for. But anyway, the collaborative work like that. So I think the UK can possibly put more, but nevertheless it is putting its money where its mouth is.
Yeah, whether that's...
Sorry, I was just going to pick up this particular point that Professor Coates mentioned earlier on about having very, very high cost, so much higher cost than we've even mentioned so far, antibiotics, and having a particular niche that you can prescribe them in, because they are only licensed for treating either a specific clinical indication or a particular resistant organism. And that, of course, solves a number of problems that I've mentioned earlier on, in that it makes regulation easier, so you're only going for a very particular area, and therefore satisfying regulatory requirements to get a drug to market is easier. And also it solves some of the issues about reimbursement because the price of the drug is so high that the company that's producing it gets some reimbursement. I think there are a number of issues, though. It's clearly very favourable for developed countries, and it satisfies, if you like, the clinical and scientific community's concern that we need to protect what we've got and avoid the development of further resistance as much as we can. So by restricting the drug use, we get around that issue. But I must say, I just wonder how that model would work for the worldwide use of the antibiotic, and in particular, given that most of the drug resistance that we see today is originated in areas of the world where they wouldn't be able to afford such high costs.
But presumably the risk is that people will just copy it and start churning it out and flogging it all over the place.
Absolutely, yes.
It depends how difficult it is to copy.
Yes, but you could copy it badly, presumably, and then still sell it. The gentleman over here.
Yeah. Robert Shaw, Chemistry, Birkbeck. I want to come back to an original statement about collaboration. I think I've been practicing interdisciplinary and international collaboration all my career. And in one of those international projects, where you were born, Michael, in Calcutta, I was giving an address to the Indian Chemical Society. I was suffering from acute diarrhea acquired in Nepal. And the head of the chairman sent out one of his minions, got me a sulfonamide, and said, whatever you got inside you, whatever in that bottle of water you're going to drink, he'll take care of it. And it did. I think what we need is an open forum where we can discuss different ideas. And I found, although I tried very hard to get a link to somebody who might be interested in my ideas in biology, very difficult. I tried about 35 years ago to find one of the medics with whom to collaborate. We didn't get very far. Jeremy Nicholson at Imperial College got a number of ideas which I thought about 40 years without producing results. Right. We need a forum where we can brainstorm and bring up. I call myself an inorganic chemist because the framework of my compounds are phosphorus and nitrogen. Robert Robertson, who discovered me when I was a young lecturer, said, nonsense, Shaw, you're an organic chemist. We need to get somewhere where we can discuss this and an odd idea might spark interest somewhere else.
Thank you. So the problem is that medics are not listening to chemists.
No, I think that's entirely right, but it's increasingly it's changing very quickly. I think in the clinical field there's much greater collaboration now with basic chemistry. Also other specialties that you haven't mentioned in terms of the epidemiologists, the veterinary scientists as well, who I always find absolutely fascinating to talk to. They have a completely different insight to infectious diseases and microbiology that I do, because they see a different range of organisms and different antibiotic resistance issues. But I agree entirely that because the knock-on effect from this sort of silo working, if I use the cliche, is that often some of the research and the funding streams follow those silos, whereas we do need to think across all of them. And Chris, I'm sure, will be able to talk at much greater detail than me about that.
Well, I absolutely agree with that view. We need to get people to work together if it's going to be done in the academic community. And just another little ray of hope here, that's starting to happen. So in the neglected diseases field, of so-called and tropical diseases, for example, malaria and tuberculosis, there are public-private partnerships that are working there. For example, GlaxoSmithKline, to give the pharmaceutical industry, which always gets a bad press, has invested a lot of money in a facility in Tres Cantos in Spain, where they're doing, as far as I'm aware, not-for-profit research to develop new compounds for malaria and TB. And maybe we need to take that type of approach and apply that to the resistance problem. In fact, I believe if it's going to be done in the academic community, there is no choice. We have to do it in an open way. I'd even suggest that we consider doing it in a patent-free way, because patents and lawyers inhibit the communication. And the fact is, one of the problems, if you like, in the antibiotic field is that the antibiotics are really cheap, and that's because they're off patent. So one solution would be to extend the patent lifetime, and maybe we should do that as well for the new high-risk research. Another approach would be to attempt to do it patent-free. And many of the so-called generic companies make a very decent living off antibiotics without any patents on the structures. So if, from my perspective, if we're going to do it in the academic community, perhaps in partnership with some of the industry, we could consider doing it patent-free.
Barry.
On the patent side, there's a very interesting debate on the internet. I used to think, well, fine, just give a longer patent. But being provocative, another argument is that if you extend the patent, these companies will be selling as much as they can before the patent runs out, even longer. So it's not quite as straightforward as you'd think.
But I'm talking about new research to make new antibiotics.
Trying to encourage.
Okay. Question over there. Yes.
Research.
Thank you.
Esther Torrett, University of Cambridge. I've got a couple of questions, if that's all right. The first one is, where do you think our new antibiotics are going to come from? Are they going to come from natural products, existing compound libraries, or through sequencing of bugs to identify novel targets such as glaqueline? So that's question number one, I think probably to the chemist. And then question number two is, one of the things that we really should have learned through clinical practice is that giving antibiotics on their own is probably not a brilliant idea. So you've learned from HIV and TB and malaria that, if you introduce one new drug class at a time, obviously drug resistance develops. And do you think that we need to rethink our whole approach in terms of the treatment of infectious diseases? It's probably slightly controversial, and that's probably to Nick.
So first of all, where the drug's going to come from?
So a comment on that. I think whatever works best. I think the barrier between natural and unnatural compounds has gone. In fact, the penicillins that are used commercially are not really natural compounds. They're modified structures produced by modified fermentations. So we do whatever works best. I totally agree with the point that Barry was saying earlier on, that we need to apply modern medicinal chemistry methods to antibiotic discovery. And I contend that if a tiny fraction of the money that had gone on cancer had been spent on antibiotics, we just wouldn't be in this situation now, just 5 or 10% of it. What was the other question? Genomics, DNA sequencing. Well, it hasn't been very successful so far in terms of developing new antibiotics, and arguably in terms of medicines in general. It will pay off in general, I believe it will. Do we need that to develop new antibiotics? I don't think so, but it might be helpful.
The other question was about whether we should be hitting them with 2 or 3 things rather than just one thing. That trying to pick them off one at a time is...
Okay, so this is a rather subtle target, but arguably all the major antibiotics hit more than one target anyway. So certainly all the penicillins hit more than one target inside the bug, and that may be the case for the other classes as well. So this is a rather technical argument for doing screens on cells and in animals, as well as against isolated targets.
Barry, so the solutions come from the natural world, or...?
You've got to try everything. I mean, Hong Kong started, I think, 10 years ago to look at all of the compounds they're using in alternative medicine. It's not going very well, I understand. They're having a lot of trouble. There are literally hundreds of compounds in there. And they're not quite sure which are toxic, which are therapeutic, and which are therapeutic because there's a combination between them. But I think we've still got to look at the Amazon forest. I'm sure it's got lots of things in there which we will discover. Resistance to it now, I know. So we've got to try everything. And there was another question, wasn't there? Yeah, so in theory, I think combinations should be, for certain types of infections and certain types of units, advisable. But a systematic review recently, actually, wasn't it, showed that single therapy seemed to be just as good as combined.
Yes, I was going to say, we have to be slightly careful. Of course, this has, as Dr. Torek has mentioned, quite a history in that in the early days of tuberculosis therapy, it was appreciated that if you used a single agent, then drug resistance developed incredibly quickly, and that's why we used 3 or 4 drugs for a long period of time. But theoretically, if you'd use more than one agent, you could either increase bacterial killing, that is the 2 drugs that act synergistically, and better treatment of the infection is clearly a good thing, or potentially, as was mentioned, reduce the risk of emergence of resistance, partly by killing the organism quickly, but also if the resistance develops to one drug, then at least you've got another one to mop it up. In some situations, this, I think, it is useful. However, we have to be slightly cautious in that 2 drugs are more toxic than 1 drug, and there is, of course, the potential for antagonism between the 2. So in the early days of treatment of pneumococcal pneumonia, with people tried combining penicillins with tetracyclines, which drugs that were available in those days, and that is one of the first examples that we have of clinical antagonism, that the mortality was actually higher in the group of people who are treated with the 2 drugs. So in principle, yes, it's something that needs further exploration, but it's not something that we should just leap at and do in an uncontrolled way.
If you combine a static drug with a cyclical drug, it's not a good idea.
Yeah.
And it depends on the infection. The other thing that you might want to know is about cycling of antibiotics on intensive care unit. That's a whole area which...
You look as though you want to come back on that one.
Surely one way to protect that is to use it in combination, because basically you're just adding in a new drug to a failing regimen, potentially.
Because weren't you suggesting, Chris, this new antibiotic which you were getting excited by was a kind of combination?
Yeah, the AstraZeneca compound is an antibiotic plus a compound that protects the antibiotic as well. But I absolutely agree, and this may be a regulatory issue which I don't know about as well. There could be a lot more work done on combinations. The work that is there on combinations, I think suggests they're extremely good medicines, with Augmentin will be one example there.
Yes.
If I may, it's John Watson again. Nobody's mentioned diagnostics, and in this utopian world of 20 years' time, a patient presenting to the doctor would have a test would not only tell the doctor what the drug susceptibility of the bug is that they've got, so that they could get very specifically the right antibiotic for that bug and nothing else, but actually also tell the doctor whether they've got a bacterial infection or not, and if not, give them no antibiotic at all. And I wonder whether the panel feel that in practice this is an area which will contribute positively to reducing the problem we're discussing.
Yeah, okay.
Yes, certainly, I can talk to that. And in my introduction at the beginning, I mentioned that most antibiotic prescribing that we do in hospitals is empiric. That is, at the time that we write the prescription, we don't know what the bug is, and we don't know whether it's resistant or not.
And you said you often get it wrong.
And the risk is, therefore, that you get it wrong. Though we think we're pretty good at guessing. But yes, accurate diagnostics is a key issue, as Professor Watson said, for not only deciding whether or not someone actually has a bacterial infection in the first place, because if you can exclude that and say it's likely to be a virus, they don't need antibiotics at all. Or, ideally, if they do have a bacterial infection, narrow it down and tell you, ideally, within the time frame that you're thinking about writing the prescription, exactly what they've got. Now, the conflict here is that at the moment, although the technologies have advanced extremely rapidly, even over the last 2 or 3 years, particularly with molecular diagnostics, the evidence suggests that, particularly for the most severe forms of infection, the time between the initial presentation of a person to hospital with severe sepsis and the first dose of antibiotic is absolutely critical. And hence we have an initiative called the Sepsis 6, where one of the 6 things that we have to do to reduce mortality is to give the first dose of antibiotic within an hour of the patient presenting. So that means, in the ideal world, we'd need a diagnostic test that could tell us within that time frame what the patient has got, and ideally the antibiotic susceptibilities as well, which sounds at the moment pretty pie in the sky to a specialty where most of our diagnostics are still performed by plating specimens onto a petri dish and incubating them overnight.
So it takes currently how long? So
to get an organism on a plate and then confirm its antimicrobial susceptibility is usually 48 hours. Right. Clearly not a time frame to influence immediate antibiotic management. But that is the whole point of the current antibiotic prescribing strategy that we have in the UK and also in the States and other countries too, which is to give an immediate dose of antibiotic, a broad spectrum, a best guess treatment, and then narrow down to something more focused as soon as you know what the cause of the infection is. But that's getting away from Professor Watson's main point, which is what we need are rapid diagnostics. And I think 2 years ago, I would have been very pessimistic that we would have something within my working lifetime. But as I say, the changes in molecular diagnostics, in other diagnostics such as, for example, mass spectrometry used in routine laboratories now to identify organisms very, very quickly. The way that they've been introduced in really a period of months gives me quite some cause for optimism. I think that within not too long, we will have point-of-care tests that will be able to give us much more information.
We do rely on culture before we can identify bacterial organisms, but when we get to a stage where we'll be able to sequence directly from clinical specimens, I think that will be a major breakthrough.
2 questions. Gentleman over there and over there. Lady over there.
Hi, okay. Thanks. Stephen East from Evotech. I'm a medicinal chemist, so I share Professor Schofield's optimism that we can solve it if sufficiently incentivised. But my question is actually back at Dr. Nick Brown. A specific patient population that could be diagnosed is one that comes in for treatment, whether it be an operation or something. So is there a case that for that population you could diagnose up front before they're admitted to hospital to understand what their susceptibility to certain organisms is likely to be in case of a problem post-operation?
Well, there's a very good example of where that is already being done, of course, and that's with MRSA. That currently all patients admitted to hospitals in England for elective surgical operations are screened for carriage of MRSA, and if they're coming in for a procedure where antibiotic prophylaxis is given, then that prophylaxis will be amended according to that result. Of course, we've now got to a stage where MRSA, fortunately, is much, much more uncommon than it was, and such a strategy is being revisited at the moment, but I mean that is clearly an example of where that is done. Other analogies with urinary tract surgery, it is also universal practice to screen patients for the carriage of organisms in the urine to look for organisms that might be resistant to the antibiotic prophylaxis. So yes, it can be done and is being done at the moment.
Another question over there. Sorry,
your question is also relevant to the vaccine strategy, because if you're thinking about who should have this vaccine, you're thinking what operation or what condition they've got, what intervention they're going to have in that hospital, what are they at risk of getting. So if I were having a hip replacement and there was a successful Staph Aureus vaccine around, I would quite like to have that. If I was in a long-term care facility and I know there's a particular problem with Clostridium difficile, I would tend to have my C. diff vaccine each year rather along the lines of my flu vaccine each year. So tailoring the approach to the risk.
Okay.
Hi, Alex Ferguson from the National Cancer Research Institute. My question is for Chris. I was wondering whether you think there'll be a day that we discover a completely new class of antibiotics that are a lot simpler than the ones we have at the moment to synthesize. Is the structure of an antibiotic really integral to its function? And my second is just more of a general comment, really. The NCRI is a partnership of 22 different cancer charities that spends a lot of money on cancer research, and I can't think of one charity that funds research into antibiotic resistance.
Okay, so the first thing to say is there are, just correct you on that, there are some charities that fund work into antibiotic resistance. So in addition to the research councils, the Wellcome Trust has put a lot of money into antibiotics as well, including via the CD Drug
I think the point
project.
is they aren't charities in the established sense that we think of a lot of the cancer charities, cancer research. The public have not got behind it, let's put it that way.
Okay, yeah, perhaps not so, but maybe Anthony could comment on this as well. So, There's a British Association for Antimicrobials that does a lot of good work as well. But it could, I agree, they could be much higher profile as well. What was your second? So I question the drive for novelty in terms of antibiotics. But I think there must be other classes of antibiotics to be discovered. And there may well already be good antibiotics sitting there. And the golden age of natural products was the sort of 1950s through to the 1980s. And really, we stopped doing natural product science then. And some really interesting, I can see a few people nodding in the audience, some really interesting structures there that weren't pursued by the industry. In part, they haven't been picked up now because of patent issues. Once they've been described once, companies are reluctant to pick them up again. So absolutely, I believe the case. And I believe that modern organic synthesis, chemistry, can solve any synthetic problem we have with antibiotics. We could do penicillins, we've done carbapenems, we've done tetracyclines, erythromycins. We can do it. Chemistry has the power to do the chemistry bit of antibiotic science. Will it be simpler compounds? Yes, probably. Chloramphenicol is a very simple structure as well. It's not so widely used here, and drugs don't have to be complicated to be useful. So yes, there are new mechanisms to be discovered, but to come back to my original point, if this is an immediate threat to society, which the Minister of Health has identified as such, what would we do? And in my opinion, the first port of call would be the validated targets, if it was a wartime situation. Cell wall biosynthesis and ribosomes, gyrases maybe, and one or two other targets. We wouldn't need any others in the short time frame.
This is actually, sorry,
speech.
I was going to say...
it's not a dream. It can be done, right?
We're running out of time.
but we just need the investment from the taxpayer to do it.
We're running out of time, so what I really want you to do now, each in turn, is to make your sort of passionate belief about the future. Where are we going? Are you optimistic?
Shall I start?
Yeah.
Yes, I am optimistic. But I think that we have to be realistic and say that it takes between 5 and 15 years to get a drug from completely nothing to market. And that's assuming we can discover it now. So realistically, it is going to be 5 years before we get a completely new antimicrobial agent available for use. And Chris is right that there are a couple of things that are currently in the sort of final stages of drug development and regulation, but with perhaps one exception, they're not going to be earth-shattering in terms of covering all the resistant bugs that we've got problems with at the moment. So I think we do need to look very critically at the whole production plant, if you like, for antimicrobials and the way that we do it. I agree entirely with Chris that the funding model is not right at the moment. And how much we can change that as a single country and how much of it would need to be an international collaboration, I'm not quite sure at the moment. But certainly I think the impetus to do something has increased exponentially over the last 18 months or so. And so I certainly get the impression that people who can do something are now very interested in doing something about it.
Barry?
I'm very positive. I've seen the effect that Sir Liam Donaldson had globally via the WHO to improve patient safety, and I think Dame Sally could do the same thing, driving this antimicrobial resistance strategy. It's not just one thing, all 7 things are important, and organisational behavioural change, which we haven't mentioned, is also vital too.
Chris, a final word. Hurrah for the chemists.
Well, it's absolutely not just about chemistry.
Yeah.
I think in this particular field, there's a really good spirit at the moment, particularly in the UK, between chemists, biochemists and clinicians as well. I think it's something we have been world leading at. We could be world leading at again. But what it really needs is the investment, the real investment. And in my opinion, that's the NHS is the right vehicle to fund that. The research councils and the Wellcome Trust have been great at doing the basic science. Fantastic at keeping the flag flying, and many people in industry as well, particularly smaller companies. But ultimately we need large-scale taxpayers' investment, and this could be something that the UK leads the world at.
Indeed, and possibly somebody should be creating right now in this room the next big charity, which is the equivalent of the Imperial Cancer Research Fund. Thank you very much. That's 8 o'clock. Could you please thank the panel for a very lively discussion? Thank you. 8 o'clock. What happens next?
So thank you very much.